Reactome is a free, manually curated knowledge base that provides a user-friendly platform to access and analyze information on molecular mechanisms of reactions and pathways. It provides highly detailed pathway diagrams of molecular processes and descriptions, available via the Reactome website, and to download in standard formats such as biopacks and SBML. The basic unit of Reactome is a reaction, which is defined as any molecular event, including binding, phosphorylation, biochemical catalysis, molecular transport, and spontaneous molecular events. These reactions can involve any biological molecule, but are typically proteins and small molecules. A pathway is defined as a series of connected reactions, with each individual reaction considered as a single step in the pathway. These are represented as pathways in detailed molecular diagrams. Examples of pathways include immune system signaling, the cell cycle, and apoptosis. In Reactome, we create pathways using reactions as building blocks. Reactions are interconnected based on biological relevance, resulting in the formation of pathways. The key aspects of Reactome content are quality and attribution. Pathways are written by expert biologists and independently peer-reviewed before being included in the database. Reactions always cite scientific literature that experimentally verifies the described event. These manually curated human pathways are used as templates to computationally infer equivalent pathways in other species, including most commonly used model organisms. High-throughput technologies allow researchers to identify gene regulation markers and determine gene expression, protein and metabolite levels for many thousands of molecules in a single experiment, generating huge amounts of data. Interpreting these data requires an understanding of the connections between these molecules. An effective way to do this is to integrate experimental information with well-annotated established pathway data. Reactome fulfills this need by providing free access to detailed pathways combined with analytical tools. Reactome includes tools for analyzing the pathway context of your data, mapping expression or other quantitative data onto pathways, or extending pathways with interactions from external databases. There are two ways to access the analysis submission form: either clicking the Analyze Data button on the homepage, or using the Analysis button on the Pathway browser header bar. Upon clicking the Analyze Data, an interface pops out where you can submit your data and perform various analyses. The tabs on the left of this interface indicate the different types of analyses. Analyze Gene List or Analyze Gene Expression will perform pathway enrichment analysis. Species Comparison will compare human pathways with other species. Tissue Distribution will compare pathways in various tissue types. You can select the analysis you want to perform, paste in or browse for a file containing your data, or use an example data set.